All right, welcome to our last chapter for Bio 23, Chapter 7, the control of microbial growth. All right, so to get started, we're going to go over several terms that we need to know. So sterilization is the removal of all microbial life. We see that with commercial sterilization. So this is just enough to kill the botulinum endospores. And this is, we commonly see this commercial sterilization when in the production of food. But we also see a different term used, and that's sanitation. And sanitation is different because we're not killing all microbial numbers. We're just reducing their numbers to a safe level. So sterilization is getting rid of all microbial life, whereas sanitation is just reducing those numbers to a safe amount. Just like we were talking about antibiotics, different substances that control microbes can be bacteriostatic, which is going to inhibit bacterial reproduction, or bactericidal, which is going to kill the microbes. And then more specifically, a fungicide is going to kill fungi, a sporicide will kill endospores, and a germicide will destroy all microscopic pathogens, and that's going to include viruses. Asepsis, or the aseptic technique, I'm sure those who are taking the lab are familiar with that. That is the absence of bacteria, viruses, or other microorganisms. And this is very important during surgery, or if you're working with different bacterial cultures, as you guys are experienced with. Then we also have other antimicrobial chemicals. And these chemicals will destroy pathogens, but they might not destroy them to the level of sterilization. And there's two different types based on where you're going to use them. So a disinfectant is a chemical that you will use on a non-living surface, whereas an antiseptic is something that you use on living tissue. So don't get those confused. So disinfectant, non-living surface, antiseptic would be like on your skin. So different things that will affect whether antimicrobial treatment is effective are the number of microbes. So the higher your initial population is, the longer it's going to take. So that's what this graph is showing here. So if we have a higher population to begin with, it's going to take longer to kill them all. Also, different microbial characteristics are going to affect a disinfectant's effectiveness. So whether it's an endospore or if it has a biofilm, that can affect it. The different life cycle phases can affect this, as well as different species and strains are going to have different susceptibility levels to different physical and chemical controls. Also, the environmental factors that the microbes are in, that's going to affect it. So temperature, the presence of organic material. So if microbes are in, say, blood or feces, when you put your disinfectant on that, the presence of these organic materials will affect that. So the disinfectant can be affected by these organic materials, as well as exposure time. So this is very important. So different microbes will need to be exposed to the disinfectant for longer periods of time in order for it to be effective. Start by looking at several physical methods of sterilization. The first one we're going to look at is moist heat. So this moist heat is going to denature the proteins of the microbes. One way that you can do this is through boiling. However, this can be very time consuming because some endospores require that you boil them for 20 hours or more before they are killed. So, to solve this problem, the most common way of sterilization is to use an autoclave. And autoclave is going to put whatever it is that you're sterilizing under pressure. So it's kind of like your pressure cooker, how you don't have to cook it as long because when you pressurize something, it'll cause the steam to reach a higher temperature than it normally would. So you're going to pressurize the autoclave. The autoclave is going to pressurize it, and so that'll reach a higher temperature so that you don't have to keep it there at that hot temperature for so long. So what we normally see, instead of over 20 hours of boiling, you can autoclave something for only 15 minutes at 121.5 degrees Celsius at 15 psi. And this will kill all endospores. Then another method is pasteurization. So usually, unless we're looking at the ultra high temperature, pasteurization is not necessarily going to be sterilization. You're usually just going to reduce the numbers or sanitize, right? We used that term earlier. The substance, you're just going to reduce those microbial numbers through pasteurization. So that's why your milk, the amount of microbes in your milk has been reduced, but eventually those microbes still grow, and that's what causes your milk to go bad. So the classical method was 63 Celsius for 30 minutes. Now, in the US, the most common is to do 72 degrees Celsius for 15 seconds. And we call that flash pasteurization because it's so quick, only 15 seconds. But some organisms are going to survive that. Now, another method is the ultra high temperature method, and that is 140 degrees Celsius, and that's less than a second. So that's even faster, but a much higher temperature. And this will actually sterilize the product. So milk that has undergone ultra high temperature, it's actually going to be sterile, and so you don't have to refrigerate it until after you open it and it has been exposed. Another method is dry heat, and dry heat is going to kill through oxidization. An example of a dry heat sterilization is flaming the loop, like you guys have done in lab, for those who are in lab. Another thing would be putting things in the oven. So you can sterilize them by just putting them in the oven at 170 degrees Celsius for 2 hours. So similar to putting things in an autoclave. And also incinerating something. So for example, incinerating a carcass would kill the microbes that are in the carcass. So anthrax, Ebola, etc. Another method is filtration. So filtration is going to be used for heat-sensitive materials. Like a lot of medias that have protein. So a lot of your medias have blood in them, and you don't want to denature the proteins that are in the blood. So any of these proteins. So you don't want to heat them up, but you instead will want to filter them. So you can use a filter. So I'll skip down to the fluids. So you can have membrane filters for fluids. And what a filter is it is going to have these little pores, and you have different sizes of filters. So anything that is larger than the pore size that you have, that'll get stuck on the one side of the filter, and everything else will flow through. So for example, like with the fluid filter, you're going to pass the fluid down. Here's the filter. You're going to put the fluid down through the filter. Usually you'll use a vacuum to speed the process up. It'll pull the fluid through the filter faster. But anything that is too big to fit through the pores will stay up here. Anything that is small enough. So depending on your pore size, if you want to let proteins pass through, your filter will be small enough to make sure that none of the microbes go through, but then it will still be large enough to allow proteins to go through. So pore size for bacteria. So if you just want to make sure bacteria doesn't go through, you can use a pore size of 0.2 to 0.4 micrometers. I want to have your filter strain out any viruses, it's going to have to be 0.01 micrometers, and that's generally too small to let proteins go through too. So that's it. Then we also have air filtration. And so we usually use a HEPA filter, and that will filter out any microbes that are in the air. Other physical methods of control. So low temperature, you're familiar with that with your refrigerator or your freezer. So that's going to have bacteriostatic effect. So low temperature is not going to kill microbes, but it will prevent them from growing. So refrigeration will slow it down. Freezing will usually stop any microbial growth. Also high pressure will denature proteins. Desiccation, so remember desiccation is drying it out. So dry environment will prevent metabolism of microbes. A hypertonic environment will cause lysis. And radiation, so for example, like a UV radiation, will damage DNA, microbial DNA. So those were physical methods of microbial control, and now we're going to move towards looking at chemical methods of microbial control. There's not very many chemical agents that will achieve sterility. And things that will affect this. So if they're in the presence of organic matter, how closely the chemical can penetrate into the substance that the microorganisms are on, as well as temperature will all have an effect on whether chemical methods are effective. So there's two different methods we're going to talk about where you can test the effectiveness of these chemical methods. And the first one is the use dilution test. So in this test, metal rings are going to be dipped in bacteria and then dried. And then you'll take these metal rings with the dried cultures on them, and you'll place those in disinfectant for 10 minutes at 20 degrees Celsius. And then after it has been in the disinfectant for 10 minutes, you'll remove the ring, and then you're going to place it into culture media. And then if you see growth, then you know the disinfectant wasn't fully effective and it didn't kill all of the bacteria. Now, the second method is the disc diffusion method. And if you guys were in lab, you would be doing this. So in this method, you're going to take a disc of filter paper and soak it in the chemical disinfectant. And then you place that disc on an inoculated plate. And then the disinfectant will spread, and you'll see this is zone of inhibition. So in here, all of the bacteria have died because of this disinfectant. And so you can measure the effectiveness of these chemicals by looking at zone of inhibition. So the greater area that the growth of these microbes has been inhibited, then that's going to mean that disinfectant was more effective. The actions of microbial growth, so how these control agents are going to have their effect on the microbes. So they can alter the membrane permeability of the microbes. So some examples of this would be like Lysol, iodine, phenolic compounds, and ethanol. These control agents can also damage proteins. So an example of that is bleach. And they can also damage nucleic acids. So like UV radiation and glutaraldehyde is going to have these effects on damage to the nucleic acids. And we're also going to look at these surfactants. And surfactants are not so much sterilizing, but they are more going to act by mechanically removing microbes. So when you think of this, think more like soap. So soap, the way soap functions, remember, is through emulsification. So that is going to, so these surfactants or soaps, they are going to have a hydrophilic side and a hydrophobic side. And so the one side will dissolve in the water and the other side will bind to the dirt or the grease or the microbes that like are on your hands, for example, when you're washing your hands. So soap actually acts in a mechanical action. So it's going to bind to like your skin, like your skin that's flaking off, your dead skin, the grease, and the microbes that are on your hand. So the soap will, on one side will bind to those that are on your hands, and then on the other side will bind to water. So then as you wash them off, then like the microbes will be mechanically removed from your hands. So that's what these surfactants are doing. And a couple of examples. So we have acidic and ionic detergents. And so these have an anion, and the anion is going to react with the plasma membrane of the microbes. So this is particularly important in sanitizing dairy equipment, or also like food processing centers, is where we're going to see the acid and ionic detergents used the most. And then another example is the cationic detergents. An example of this would be like dish soap. And this is more bactericidal because it's going to denature the proteins and disrupt the plasma membranes. They're particularly effective against gram-positive bacteria, but they can be effective against gram-negative bacteria as well. And lastly, we're going to finish up by looking at the ways that microbes can like fight against the actions of disinfectants. So gram-negative bacteria have porin proteins in their outer membrane. And these proteins are responsible for the prevention of the entry of disinfectant molecules. The mycolic acid in mycobacteria prevents the entry of disinfectants. Cysts and endospores are highly resistant. That makes sense, right? So they're going to be resistant to disinfectants. And also viruses. So interestingly, non-enveloped viruses are actually more resistant because the capsid or the proteins, right? This protein coat is actually going to be more resistant to chemical disruption than lipids are. So enveloped viruses, remember they have the lipid layer, the lipid envelope layer on the outside. So that's more easily disrupted by disinfectants than capsid would be. And that concludes our chapter number 7.